Hello, Clyde. Welcome back. This is our Lab 9 sheep brain dissection, which unfortunately for you will have to be done virtually. So all that we have to work with here are PowerPoints, images, and commentary. And alas, I have what I believe to be an appropriate video, which is on YouTube, and the link to that video is on the last slide. I hope that you enjoy it. Well, you don't have to grow up on a farm to know that a sheep is a four-legged creature. Because of that anatomical difference with people, the brain is laid out in more of a horizontal fashion, whereas the human brain is laid out in more of an up and down vertical type fashion. For four-legged creatures, because of that horizontal layout, the areas are referred to as rostral, meaning toward the nose. So in this picture here, that would be to your left. Toward the sheep's nose is the rostral end of the brain. To the right, that is the caudal end of the brain. Caudal means toward the tail. So you're looking then at the whole sheep brain from the side. You'll notice that there are noodles and there are ditches between the noodles. Every noodle is called a gyrus, and every ditch is called a sulcus. In the human brain, every gyrus has a name and every sulcus has a name. Furthermore, in the human brain, what we would see are clear-cut divisions between lobes. We don't really have that in a sheep brain. There are different areas of the brain that are dedicated to different services, but the delineation between one part and another is not quite as clear in lower animals as it is in humans. In the first place, what you would have surrounding this brain is a thick leather-like membrane that is called the dura mater. Down south there, right at the bottom, it says meninges. The meninges has three layers. The outermost, the layer that is against the cranium, is the dura mater. Dura mater translates tough mother. The next layer, as you go toward the brain, would be the arachnoid. Arachnoid translates like a spider. Actually, I think what they meant to say is that the arachnoid layer of the meninges is like a spider's web. Internal to those two, lying directly against the surface of the brain, is a very thin, transparent, simple squamous epithelium called the pia mater. So those are the three layers of the meninges: dura mater, arachnoid, and pia mater. Pia mater translates into tender mother. So we don't see anything here other than that one little piece of the dura mater that can be seen there at the bottom of the view. Also from the side, you can see the crossing of the two optic nerves. That would be the optic chiasm. The cerebrum is divisible by the longitudinal fissure, the gap between the left and right cerebral hemispheres is called the longitudinal fissure. We'll see that in the next slide. The gap between the cerebral hemispheres and the cerebellum is called the transverse fissure. You can see the cerebellum from the side. We'll look at it from another angle on the next slide. That area that is pointed to and referred to simply as brain stem, we will identify more specifically as the medulla oblongata. And then, alas, we have the little spinal cord. So I have two more slides that show the whole sheep brain. Let's have a look at those. Okay, nothing too difficult here. Just another external look at the sheep brain. Starting there at the left, obviously the spinal cord is easily recognized. The cerebellum has two different areas that can be seen from the outside. The middle portion that runs and kind of wiggles left and right as it goes posteriorly, that is called the vermis of the cerebellum. Vermis means worm. It's called vermis because it kind of wiggles left and right as it goes down. The other part that is labeled there, you can see those little gyri that run horizontally, run left and right. The pointer is pointed at one of them. Those are called the cerebellar folia. So you see the vermis and you see the folia in this view. Separating the cerebral hemispheres from the cerebellum, that is the transverse fissure. In our lab, when we actually do this dissection, I have the students fold the cerebellum down away from the cerebral hemispheres so that we're looking down into the transverse fissure. When you're looking down into the transverse fissure from the top side, looking down, you would then be seeing an area of the brain called the tectum. The word tectum means roof. It is actually the roof of the midbrain, and there are four structures there that can be seen when the cerebellum is folded down from the two cerebral hemispheres. So you would see the corpora quadrigemini. That means the bodies of the four twins. Well, the structures are actually called the two superior colliculi and the two inferior colliculi. We will see those later. I just wanted you to get oriented as to where they are. So if you look down into the transverse fissure, you would be looking at the roof of the midbrain. The roof of the midbrain is called the tectum. The four structures that you see there are the corpora quadrigemini, which are the two superior colliculi and the two inferior colliculi. The next pointer there is just simply pointing out the left cerebral hemisphere. Notice that the longitudinal fissure, the big gap, separates the left brain from the right brain. Longitudinal fissure. Now, the glistening clear membrane that you can see there, that is the pia mater. It is very thin, it's transparent, it is a simple squamous epithelium that covers the surface of the brain and spinal cord. The tender mother coming at you there, Clyde. Okay, much the same structures and information here, just a slightly different angle so that you can see that clear membrane that's covering the brain there that actually bridges over the longitudinal fissure. That is the pia mater. Then you see the longitudinal fissure again separating the right and left hemisphere. The vermis of the cerebellum, you can see how it kind of wiggles left and right as it goes back. The worm, and then we have the longitudinal gyri of the cerebellum, which are called the cerebellar folia. This image is taken from the video, the YouTube video that I have a link to at the end. It's a very good video, and this is a very good picture. I don't know why she has the word fourth ventricle up there in the upper right. That is nowhere near the fourth ventricle. I will point that out to you later. Let's go with the things that you should be familiar with. Start at the spinal cord. There where we come together with the cerebellum, that space between the medulla and the cerebellum. See there, medulla, See there the arbor vitae of the cerebellum? That space that's between those two, that is the fourth ventricle. So I'm not sure why she has fourth ventricle written up there in the upper right corner. That's nowhere near, and there's not even a line going to it. So the fourth ventricle is situated between the medulla and the cerebellum. So in this mid-sagittal section, you can see the gray matter of the cerebellum. What they have pointed to here is the white matter. If something is white, that means it is myelinated. Myelin is white. So we have the myelinated fibers of the cerebellum called the arbor vitae, translated trees of life. Okay, now further along here in the mid-sagittal section, two slides previous, I was talking about taking the cerebellum and folding it down away from the cerebral hemispheres so that you could look down into that gap and see the region of the brain called the tectum. The structures of the tectum are the corpora quadrigemini. The corpora quadrigemini are the two, left and right, superior colliculi, and the two, left and right, inferior colliculi. So you can see how you would see those if you folded the cerebellum down by bending the brain a little bit to look down into the transverse fissure. So there is the superior colliculus. That will become important a little bit later on when we talk about the, the sense of vision. The superior colliculi are in the visual pathway. The inferior colliculi are in the auditory pathway. We have reflexive movements in response to flashes of light in our face. That is the duty of the superior colliculus. It is a reflex movement away from something coming at your face. You don't have time to think. You reflexively turn your head away from something coming at your face. In a similar way, the inferior colliculus, being in the auditory pathway, that coordinates a reflexive turning of the head away from sounds. So if you hear something coming at your head, you also reflexively turn away. So the corpora quadrigemini, located in the tectum of the midbrain, the two superior colliculi in the visual pathway, the two inferior colliculi in the auditory pathway. Okay, another obvious structure that is pointed to there, we will have to go into greater detail than this. That is the corpus callosum. The corpus callosum contains projection fibers that connect the left cerebral hemisphere to the right cerebral hemisphere. We only have 3 locations in the brain where the left hemisphere connects to the right hemisphere. The corpus callosum is the primary one. There is also an anterior commissure and a posterior commissure. They connect right and left brain, but the corpus callosum is the primary projection fibers from one side to the other. So you can see the corpus callosum labeled there. Out front, you can see that it bends down. That is called the genu of the corpus callosum. Genu means bent knee. Well, you can see where they got that name. It looks like the knee is bent. So that is the roof of an open space. The open space is called the lateral ventricle. So it is not labeled there, but you can see there is a membrane. The membrane is called the septum pellucidum. It separates the left lateral ventricle from the right lateral ventricle. That open space also has a floor that is labeled there. The floor of the lateral ventricle is called the fornix. The fornix reaches down all the way to the area of the brain that we will call the hypothalamus. Notice there the thalamus. There is actually a left and right hemisphere to the thalamus. The space, the very slender space there between the thalamus and the fornix is called the third ventricle. So see the relationship there between the thalamus and the hypothalamus. It is not labeled there, but between the hypothalamus and the pons, that area of the brain is called the midbrain. Notice that the superior and inferior collicula sit at the roof of the midbrain. So that space there that's not labeled between the pons and hypothalamus, that is midbrain. Its roof has the tectum, which has the corpora quadrigemini, the bodies of the four twins. Okay, maybe another label, maybe another image will allow for more detail than what I've shown here. I think it might be a good idea for you to print off this particular image and write in the names of the structures that are pointed to here. I will call them out. I don't want to take the time to try to type them all in myself, but I can identify them and you can write them down and print off the image yourself. This is pretty well detailed. There are more things to be seen here than are labeled, but this is not too bad. So let's just start with letter A. The pointer is not really, it's kind of between two structures there, but I can tell you what both of them are. So look at where pointer A ends. Well, I just named that thing a minute ago, the genu corpus callosum, the bent knee. I don't think that that's what they're trying to point to. I think that it's intended to point to the noodle, which is just on top of the genu. It's kind of pointing to the space in between, which I don't know why they would do that. So look at the noodle that sits just to the left of the genu. Genu is the bent knee of the corpus callosum. That noodle is the cingulate gyrus. We will come across the cingulate gyrus again later when we talk about limbic system structures. So that noodle that is right on top of the genu corpus callosum, that is the cingulate gyrus. So I think A is probably pointing to that. Can't really tell if they're wanting the corpus callosum, genu corpus callosum, or the cingulate gyrus. Don't really know. Okay, go to letter B. That is the membrane that separates the right lateral ventricle from the left lateral ventricle. The membrane is called the septum pellucidum. Well, you know that septum means wall, pellucid means almost clear. So it's the almost clear wall that separates the left and right lateral ventricle. Letter C is pointing to the corpus callosum. You shouldn't have a problem recognizing that. Those are the projection fibers that connect the left and right cerebral hemispheres. Okay, letter D, that is the floor of the lateral ventricle. We call that structure the fornix. Go to E, that is pointing to the space between the thalamus and the fornix. You can see it, it's black, it's open space there. That is the third ventricle. It is in the walls of the lateral ventricles and the third ventricles that we have a structure called the choroid plexus. The cells of the choroid plexus contain those ependymal cells. Ependymal cells are the cells that make the cerebrospinal fluid. So the lateral ventricles, the third ventricle, and later the fourth ventricle, and the cerebral aqueduct that connects the third and fourth ventricles, they are always filled with cerebrospinal fluid. Letter F, that is pointing to the pineal gland. The pineal gland is the gland that makes melatonin. We will talk about that when we get to the endocrinology chapter. Okay, moving to letter G, that is just posterior to the pineal gland. That is the superior colliculus. Notice that it sits right there where you would see it if you folded the cerebellum down away from the cerebral hemispheres. That area is the roof of the midbrain, the tectum. So that is your superior colliculus being pointed to there. Letter H, that is the cerebral aqueduct. Cerebrospinal fluid has to drain from the third ventricle to go down to the fourth ventricle, and that connection between the two is the cerebral aqueduct, which is being pointed to by letter H. I, that is the inferior colliculus of the tectum of the midbrain. Letter J, that is the white matter of the cerebellum, which we call the arbor vitae, trees of life. Going down to K, that is the medulla oblongata. Just above that, actually between the medulla oblongata and the cerebellum, notice that the cerebral aqueduct flows right down to there. That is the fourth ventricle. The open space between the medulla and the cerebellum is the fourth ventricle. Notice how the cerebral aqueduct runs right down to the fourth ventricle. That same opening will continue down into the spinal cord. In that location, it would be called the central canal of the spinal cord. So K is the medulla oblongata. Move along to letter L. That little pot-belly shaped area of the brainstem is called the pons. Letter M, that is the midbrain. Notice that the midbrain has a roof. The roof is called the tectum. All right, next letter, letter N. Well, we are in the area of the brain that is called the hypothalamus. Notice that it is just below the thalamus. Well, the thalamus is letter Q. Well, look at it there. All right, back to letter N. That little mass of cells that's located there in the hypothalamus, that is the mammillary body. It is part of the hypothalamus. Letter O, that is the stalk of the pituitary gland. The stalk of the pituitary gland is called the infundibulum. It connects the hypothalamus to the pituitary gland. Next letter is letter P. That is the optic chiasm, the location where the left and right optic nerves cross. Letter Q, that is the thalamus. And letter R, that is the olfactory bulb, the olfactory nerves attached to the olfactory bulb, which becomes the olfactory tract, which goes to the olfactory cortex. So we will talk about that when we get to the olfactory pathway in the senses chapter. Well, I don't know if you got anything out of that or not. So do what I said. Just print out a copy of this and write down the names yourself. There is a certain bit of educational magic that goes with simply writing things, not typing it, not saying it, just writing it down with a pencil. So print out the image and write down the names that I gave you. Okay, this image is also from the video that comes up at the end of the PowerPoint here. And Scientific Cindy well, she's only going with the easy to identify and obvious structures. Uh, the undersurface or the ventral surface of the brain is incredibly busy. There are many more things here to be identified than what she has pointed out. But since we don't have a hands-on lab where I can show you those things, I thought, well, this perhaps would be best to just keep it simple. So starting at the far right there, that is not the olfactory nerve. Those are called the olfactory bulbs. The olfactory nerves spring off of the olfactory bulbs and go into the nasal mucosa. You don't see the olfactory nerves because they get torn off when the sheep's brain is taken out of the sheep's head. They are very small and very fragile nerves. So the actual olfactory nerves are not seen. What you see is the olfactory bulb. She's got that pointed to. They continue back to the temporal lobes where we have the olfactory cortex for perception of the sense of smell. All right, so anyway, just go with olfactory bulbs for those two things that she has labeled olfactory nerve. Now, moving back from there, we have the crossing of cranial nerve number 2. Cranial nerve number 2 is the optic nerve. So we have a right eye, we have a left eye. Well, the actual optic nerves have been cut off, so you only have just a little bitty stump of the optic nerve there, right and left. The right and left cross, so you can see where the right and left optic nerves cross. That is the optic chiasm. Posterior to that, the continuation of the optic pathway, that is called the optic tract. So coming to the chiasm, those are called the optic nerves. At the crossing, that's called the optic chiasm. Posterior to the crossing, those are called the optic tracts. Notice immediately posterior to the optic chiasm, you see a little bitty hole. That little bitty hole is in the infundibulum. The infundibulum is the stalk of the pituitary gland. So we have to have things that move from the hypothalamus into the pituitary gland. The hypothalamus makes hormones that it stores in the pituitary gland. So there's a little hole there in the infundibulum, and that is immediately posterior to the optic chiasm. Now, immediately posterior to the infundibulum, there is a little whitish colored mound there. Well, that is the mammillary body. There are actually two of those, a left and right mammillary body. Those are part of the hypothalamus. Okay, oculomotor nerve, cranial nerve number 3, comes directly off of that white V-shaped mass. The white V-shaped mass of the brain there is called the cerebral peduncles. The oculomotor nerves, left and right, come off of the cerebral peduncles of the midbrain. Cranial nerve number 4 comes out of that ditch that is left and right of the cerebral peduncles. If you get a good look at those cerebral peduncles, they are white colored and they're V-shaped. The right side, left side kind of makes a V shape. The trochlear nerve, cranial nerve number 4, comes out of that ditch that's to the left and right of the cerebral peduncles. Just to the left of that, or just posterior to that, you see the pot belly. Well, that pot belly is the pons of the brainstem. We have the big doo-wop-a-daddy trigeminal nerve coming off of that pons there. Notice how big that rascal is. Well, trigeminal means 3 twins. It's a big nerve, and we will talk about it in great detail when we get to the cranial nerve section. A little bit posterior to that, also coming off of the pons, cranial nerve number 6, the abducens nerve. Now we do have cranial nerve 7, 8, 9, 10, 11, and 12. They are not labeled here. Scientific Cindy, I guess, didn't see the need for labeling the remainder. And since we don't have a hands-on lab where I can show you those things, I won't even try. So that'll be all we're going to do for the cranial nerves here. I can promise you it will get more detailed when we study the human brain. Okay, this is a view of the sheep brain. If you take a knife and you cut a frontal section right at the level of the optic chiasm. So that's what's been done here. So it is a frontal, otherwise called a coronal section. So when the cut is made right at the level of the optic chiasm, then you're able to see some very important structures here. So see if you can get oriented. Since it is a frontal section, we have a left and right part. Notice that the corpus callosum connects the right hemisphere to the left hemisphere. Notice that we have the left and right lateral ventricles just under the corpus callosum. The membrane that separates the left and right lateral ventricles is the septum pellucidum. The thalami, the 2 hemispheres of the thalamus, are the masses of gray matter situated there just below the lateral ventricles. So get yourself oriented here. What you would be looking at here is toward the eyes from the back. So you're behind the sheep's eyes and you're looking toward them, starting at the level of the optic chiasm. So that area of the brain in the human we would call the frontal lobes. Sheep don't have frontal lobes, but that's toward the forehead in the human or toward the nose in the sheep. Okay, so get oriented by finding the corpus callosum connecting the right and left hemispheres, the left and right lateral ventricles, separated by the septum pellucidum. Okay, the optic chiasm is also labeled there. The other labels I put in myself. So notice that we have gray matter, that's colored kind of beige or brown, and we have white matter. The white matter is white because myelin is white. So notice that the white matter originates way out in the cortex. Well, at this level of the brain, right at the optic chiasm, that part of the frontal lobe would be called the motor cortex. That's where the notion to make a motion originates, those pyramidal cells of the motor cortex of the frontal lobes. So those fibers are myelinated, they're white. If you trace the white matter south from the pyramidal cells out in the cortex, that tract is called the internal capsule. Notice that the fibers of the internal capsule loop around left and right of the lateral ventricles and the thalamus. I have also pointed out here 3 masses of gray matter that are situated just lateral to the internal capsule. Also, on the left side, another mass of gray matter, the caudate nucleus. All 4 of those masses of gray matter taken together are called the basal ganglia, probably better called basal nuclei because ganglia are only in the peripheral nervous system. So they are still called the basal ganglia in textbooks, but the better term would be the basal nuclei. They're located in the brain. So these 4 masses of gray matter, the caudate nucleus, the putamen, the globus pallidus, and the subthalamic nucleus, those are 4 areas where the notion to make a motion is coordinated. So if we don't have the input of the basal nuclei, then our movements are uncoordinated and meaningless. They become just basically jerking motions of the limbs. So we have the input of the basal nuclei, the caudate nucleus, the putamen, the globus pallidus, the subthalamic nucleus. They feed into the internal capsule. The internal capsule is the motor pathway, and it will later travel through the medulla oblongata as that mass of tissue that we call the pyramids. Consequently, the pyramidal cells of the motor cortex are going to cross at the decussation of the pyramids when they get to the medulla. So this is a pretty picture of the basal nuclei, the internal capsule, the motor cortex, where we initiate our intentions to move. Okay, this is the link to the scientific Cindy video that I've taken a couple of images from. Uh, what she is doing in this video is what you would have been doing had you been in lab. She is a little bit short on identifying things that I would have had you identify, but you really can't get, well, they just don't have teachers like me on YouTube, okay? So what you get then is scientific Cindy because the coronavirus kept me and you from getting together and doing the real thing. So anyway, watch it and learn from it. It is what you would have been doing if we had had this lab in person. Over and out.